Folks, so we're moving on to looking at how all the stages of the cell cycle are regulated. And the first thing I want to look at already is a little bit of coordination between growth and progression through the cell cycle. So as we think about progression through the cell cycle, we think about G1, S, G2, and M, we see variations observed cells to cells, tissues to tissues, in the overall length of the cycle, and the relative event length of time that cells spend in various phases, and then how closely mitosis and cytokinesis are coupled. And so you bear in mind that the cell cycle is regulated to meet the needs of the cell type, and in the case of single-celled eukaryotes, the organism. So at one end of the extreme, we have cells that divide continuously to replace cells that are constantly lost or destroyed. For example, cells involved in sperm formation and stem cell populations, for example, continuously dividing to replace cells that are lost. At the other extreme, we've got cells that are extremely slow-growing, it's very slow-growing tissues, or even some, for example, mature nerve or muscle cells that don't divide at all or divide very, very, very infrequently. So most of the variation time that we see in cell cycle length is based on the length of G1. Now there is some variation in S and G2. I don't want you to walk away thinking that there's no variation in S and G2 lengths, but the majority of the variation is in G1 with small variations in S and G2. So cells that divide very, very slowly may spend days, months, or even years in an offshoot of G1 called G0, where they just stay in a kind of quiescent phase, ready to divide if they need to. Whereas cells that divide very rapidly may have a short G1, or in some cases they may skip it. An extreme case of that is the early embryonic divisions you see in, for example, frog eggs and sea urchin eggs, where for the first several divisions they just have alternating rounds of mitosis and S phase and cell division, S phase and cell division. So you have one big egg cell basically that gets fertilized, and then it just divides with no intervening growth phases, and it just becomes a mass of smaller cells. So although in some cases growth is not essential to the cell cycle, the two are generally very closely linked, with an increase in mass and volume leading to then a round of cell division. Obviously, if you just make a cell divide without growing, then it's going to get smaller and smaller and smaller. So there is a linkage between cell division and cell growth. And so we kind of, when we think about cell growth, we're really here talking about an increase in mass and volume, not necessarily cell number. So cell division gives us an increase in numbers, cell growth gives us an increase in mass and volume. So a lot of the control of growth is under the control of a protein kinase called TOR, which stands for target of rapamycin, which is a type of antibiotic. And that plays a major role in a signaling network that controls cell size, which would be a consequence of growth, and coordinates that increase in growth with cell cycle progression. So the whole network is activated by TOR, and TOR is activated in the presence of nutrients and growth factors. So absent nutrients and growth factors, TOR would be switched off. And this whole other signaling pathway, which leads to cell growth and ultimately cell cycle progression, is switched off. A cell doesn't want to be growing and trying to divide if there are insufficient nutrients for growth, for DNA replication, and to produce all of the ATP that's required for the processes of mitosis and cytokinesis. So activated TOR stimulates molecules that control the rate of protein synthesis. So this protein here, S6 kinase phosphorylates the S6 protein, which is involved in translation. TOR also phosphorylates this protein here, elongation factor 4E binding protein 1. And so this is involved in translation. And so by regulating translation, you're really regulating growth. If you're going to grow, you're going to need to produce more proteins to produce all the other macromolecule types that are found in growing cells. And so by regulating all these translation factors, you can increase the rate of translation, which can ultimately result in cell growth, which can then communicate to the cell that is acceptable to undergo progression through the cell cycle. So that's all I want to talk about here really is just a little bit on the connection between cell growth and the cell cycle. You don't need to memorize all the names of these proteins or worry about them. You should know that TOR is a master regulator of cell growth and that it acts by regulating translation, and translation is important for the production of proteins, and proteins are the macromolecule type in cell which are responsible for the production of all other kinds of macromolecules in the cells. So that's it, we'll wrap it up there. I'll be back next time when we'll start looking at regulation of the individual cell cycle phases, and then we'll move on to some checkpoints. I'll see you soon.